Welcome to another essay question that we are going to be filling out today. Of course, we always got to start with the dad joke. So the dad joke is, what do you get when you mix an elephant and a rhino? Elephino. Alrighty, so let's move on. We're going to start by describing the chemoreception of smell and taste. So I have this drawn out over here for us to be able to understand. So smell, whenever you smell something, you're going to have a nice little fragrant scent or a nasty scent, depending on what you're into. Just kidding, that's disgusting. It's going to sweep up in the nose and it's going to hit the olfactory bulb over here, which also has an axon right here. Oh, right there. The axon connects the olfactory bulb to the cribriform plate up into the brain because it is also known as the thinnest part of your skull that connects to your brain. Within this olfactory bulb and axon, we're going to have a dendritic cell. With this dendritic cell, whenever that nice breath of fresh air comes in, a ligand is going to activate this GPCR, which then that GPCR is going to activate the adenylyl cyclase, and then that's going to produce CAMP. Once it produces CAMP, it's going to open this non-specific cation channel, which allows the depolarization of the cell to then open up voltage-gated chloride channels. And action potentials will eventually be sent down the way to let the brain know that you're smelling something. We also have something called ACE2 enzyme on the end here, and it's not only in the olfactory, but it's also in the placenta. So watch out, ladies. Make sure that you're not going to lose this ACE2 enzyme. Otherwise, it can cause issues to the baby if you're pregnant. So if this ACE2 enzyme is gone, you're going to have blood pressure problems, as your blood pressure will start increasing. And so this right here is what's going to affect what's going to cause you to smell as the voltage changes throughout this dendritic cell just by the reaction of this one enzyme through the different scents as they carry ligands. Next, we're going to move on to taste. Of course, we know taste as everybody uses it, except for those COVID people who couldn't taste anything, so they ate onions thinking they're apples. What we have here is we have different taste buds. Within these taste buds, we have different like valleys, I'm going to call them, which are really where the taste buds will be located. And there are around 125 cells within these little valleys and these little caves. So we're going to take a little bit closer look at these little valleys from the taste buds. First, before we do, we're actually going to discuss what salt, sweet, bitter, sour, umami, and fat are. Fat, we're not going to focus on a whole ton because that one's still coming out in research. But we will want to focus on these others as these are still considered, these are still in our textbooks and taught. But fats may be coming soon, so keep a lookout on that as they may be entering your textbooks. Salts are activated by sodium, sweet is activated by carbs, bitter is activated by calcium and quinones, sour is activated by hydrogen, and umami is activated by glutamate. And then again, we're not going to focus on it, but fats are activated by triglycerides. We're not going to go over that pathway, but we will go over all these others. So keep up, because I'm going quick. So salt here, the sodium is going to is going to enter into the cell through an ENaC channel. Whoops. And once it comes into the cell, it's going to depolarize it, which will cause a voltage-gated calcium channel to open, which will allow the exocytosis of a neurotransmitter to react with the neuron, which will then be perceived as salt. And with this, we're going to see different levels of calcium inside the cell throughout it to reach threshold and cause different action potentials at different points or the release of glutamate. Here we have our sour. Sour is activated by the hydrogen, as we see. So those are things like your acids. So if you taste a battery, you're tasting an acid. So with that, again, it's another ENaC cell that opens up. That hydrogen is going to then flow into the cell. It's also the hydrogen is going to keep the voltage-gated potassium channels closed, but it's going to open up these voltage-gated calcium channels, releasing the neurotransmitter, allowing you to be able to taste sour. Now, sweet, bitter, and umami are going to be all within the same complex. A ligand is going to bind to a G protein coupled receptor. It's going to then the alpha subunit is going to then activate the PLC. The PLC will then allow will then work together, will then cause IP3 to be activated. The IP3 is then going to move to the sarcoplasmic reticulum, which this is the only one where you get intracellular calcium to enter into the cytoplasm. Once that enters into the cytoplasm, it releases the neurotransmitter, which then allows you to perceive sweet, bitter, or umami. Umami. You're a mommy. Just kidding. We're going to move on from there. So here we have our capsaicin, which is a nociceptor, which means it's painful. All those people out there who enjoy spice, you are just addicted to pain is really what that's telling me. Just kidding. It just means that you have a higher pain to spices, while others have lower pain receptors to spices. So this is measured in Scoville, and it's measured by the amount of capsaicin. So a bell pepper has 500 capsaicin in it, while capsaicin itself has 16 million. So the higher you go up, the hotter it gets. We also have different receptors here that I'm just going to touch on a little bit. So we have photoreceptors, which detect light. We have mechanoreceptors that detect touch and pressure. We have chemoreceptors that detect chemicals, thermoreceptors that detect temperature, and nociceptors that detect pain. The location of the action potential is in the location of the brain and the frequency, which is how the brain can interpret that signal. So there we have it, the description of the chemoreception of smell and taste. Next, we're going to describe how energy is carried by sound waves and turned into mechanical vibrations, fluid waves, chemical signals, and finally action potentials for things we hear. I'm going to combine this one because I feel like this one goes really well with describing the mechanisms of hearing, and including the part of the inner field and the hair cells produce. So just this last part and this part, I feel like should have been put together. So we're going to go over there to hearing, and we are going to talk about this entire thing, and that's going to answer those questions for us. All right, first up we have this ear. This is what a normal ear looks like for some people, but for people like myself, sometimes it looks more like this. Just because I was always told ever since I was growing up that I have big ears, so look out. It doesn't mean anything though, or at least not that I can tell, because I can hear just about the same as everyone else. With this being said, the ear is going to condense different sound waves that it hears. Those sound waves are going to enter into our external meatus, which is then going to allow, external auditory canal. I don't know why I said meatus. Which is then going to be compressed to compressed, uncompressed, compressed, uncompressed, compressed, uncompressed, compressed, uncompressed, up until it hits the malleus, incus, and stapes. We hear these in frequencies, so 20 hertz is the lower frequency, and the higher the hertz, the higher the frequency. Whenever it hits the malleus, incus, and stapes, it's going to pump back and forth and pound, 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 pound against this tympanic membrane right here, also known as your eardrum, as everybody likes to call it. Whenever this eardrum moves, it's going to move different fluids. But first, let's talk a little bit about the anatomy of the ear a little bit. So in here, you have the cochlea, which is all of this right here. So here you have your scalar vestibula, which is this outer part right here. Then you have your scala tympani, and in the middle you have your scala media, also known as your cochlear duct. As you see written here, the scala vestibuli has perilymph along with the scala tympani. As you can see, that makes sense because both are connected. So you would want to see the same fluid within the same within a similar compartment. And then in the compartment here, you have the endolymph, so that way it can perform different functions. Here at this line, you have the helicotrema, which is basically just where the scala vestibuli turn into the scala tympani. Just another thing to keep in mind is that the frequency of the hertz that you hear is going to be the pitch. The amplitude of the hertz that you hear is going to be the volume. And then talking about this little green part right here, we have the basilar membrane. I'm going to talk a little bit more about that here in just a second, but first I want to say we're going to come back to how this liquid is moving in just a second whenever the tympanic membrane vibrates. So moving back to this basilar membrane right here is if we chop it up by looking at it in this direction, we are going to see this image here, as if you flip this sideways and you can put it in. These fibers here are going to be short and tight. That will allow you to be able to hear high-pitched sounds, while these fibers over here are going to be long and a little bit looser, so that will allow you to hear low-frequency sounds. With this here, as age continues, you start to lose the different fibers here that allow you to hear high-pitched frequencies. So whenever your grandpa says, "Speak up, I can't hear you," rather than speaking up, something you might want to do is just speak in a lower tone, so that way they can hear you over here. Because these fibers have not yet been destroyed, unless your grandpa's just deaf. Then you may just want to get a new grandpa. Just kidding, don't do that. But moving on is, with that is whenever the waves come in contact with it, it's going to cause these to vibrate. It's going to do that with resonance. Some vibration will resonate with these, while other vibrations will resonate with the longer ones. So just pay attention to resonance, because they're not going to resonate completely equal. Alrighty, we're gonna chop this again by looking at it in this view. And with this being said, this is what it's going to look like if you're looking at it at that angle. Up here, just remember where we are. Up here, you have your scala vestibulae. We just amplify the scala media so that way I can fit everything in my drawing. And down here, you have your scala tympani. With this being said, some important structures to talk about is this tectorial membrane right here that's going to be moved by the hair cells, and these hair cells right here. Sorry. All right here. Let's not duplicate that. And so these hair cells right here are your inner row hair cells. These hair cells right here are your outer row hair cells. I'm just talking about a little bit of tissue potential. Your tissue potential is going to be 80 millivolts. So you're going to have negative 80 in the scala tympani, while you're going to have positive 80 in the scala media. You also, inside your hair cells, you're going to have negative 40 across each hair cell. Now, before we get more into how this causes action potentials and what's going on, first we're going to talk about how the chemical gradient is established. We're going to focus a little bit on this stria vascularis right here, which is going to demonstrate to us how that chemical gradient is established. Here we have blood vessels that is supplying nutrients and different oxygen to the end of this stria vascularis. It's going this stria vascularis is going to be made up of two different sides, two different membranes and cells. And so this here is going to have sodium-potassium channels, allowing sodium to come out and potassium to come in. That potassium is going to be important, so pay attention to it. Then we're also going to have channels that pump sodium back into the cell, allowing more potassium to come into the cell. Then you also have to neutralize the charge gradient, so you're going to put two chlorines into the cell. Well, once this happens, though, you have potassium leak channels over here that are going to allow the potassium to leave the cell. Once this happens, the chlorine will also leave the cell, so that way it can establish a proper charge gradient within the cell. So we're going to have a buildup of potassium in this space right here, and then we're going to have to go through the cells again. With this happening, it's going to cause more potassium buildup inside, and the same thing happens. Potassium leaves, chloride leaves, potassium leaves, chloride leaves, and then we have more buildup of potassium in the endolymph over here in the scala media. So that's why we're going to have more potassium there. Now, pay attention to that, because that's going to be important in the hair cells over here. Alrighty, so let's talk a little bit about those hair cells. These hair cells are going to look similar to this. They're going to have mechanically gated potassium channels that are linked together to the fiber next to it, which then, when moves, opens up that voltage-gated potassium channel. It's also going to have voltage-gated calcium channels over here that will allow the exocytosis of a neurotransmitter, which will be picked up by these signals of the brain, so that way the brain can respond properly, interpret properly, and respond properly. We also have these proteins over here called prestin, which are broken chloride cells. Sorry, I forgot to add in the chloride part. Sorry, broken chloride channels, in which meaning it won't allow chloride out, but these will still move in accordance to the different voltage, the change in voltage that is being accumulated whenever the hair fibers move. Alrighty, so first, before we talk about the opening of these and the changing of prestin, we are going to talk about this green part right here, which, as we know, is what? That's right. Ignore that message. That's right. It's the basilar membrane. So when that basilar membrane vibrates, it's going to move these cells a little bit to start to pick up on sound that it's like, oh, I think I'm hearing something, but it's not really enough for you to be able to hear it super well. So that's why you have this concentration gradient of this chemical to amplify this signal. Whenever this prestin is stimulated by the change in voltage, it's going to increase and decrease really fast or really slow, depending on the frequency, depending on the voltage amount. But with that being said, is whenever it does change, increase and decrease, it's going to vibrate this tectorial membrane. Whenever this tectorial membrane is going to cause different waves within this endolymph, and whenever it causes waves in the endolymph, it's then going to cause these hair cells to then move, which, again, as we know, whenever the hair cells move, it's going to open up potassium channels, allowing the release of a neurotransmitter, which then the brain receives signal. Whenever the brain receives the signal from the inner row, that's when we hear different things. So you can pick up on what I'm saying now. You can be like, oh, this guy's dad jokes actually aren't funny, or they are funny, as we all know is true. And over here, the brain is going to be able to stimulate appropriate responses with these hair cells. Oops, let's undo that. With these hair cells over here, so that way it can tell you whether something is important. So let's say that you're not paying attention to background noise. This is going to cause um, the inside of here to need a higher a higher change in order to pick up on the signal, because it's like, oh, it's background noise, I don't care. So it increases the amplitude that is being needed in order to pick up on what is being said, while other things that you're paying attention to, it has the appropriate amplitude needed. So all those who weren't listening to my dad joke at the beginning, your amplitude was really low to that. Well, sorry, your threshold for that was really high for the amplitude. So I guess I just needed to say but if I said your name, then it would be increased and you would be listening more to my dad joke. So that way you can be enlightened for the rest of your night. And so that is how you are hearing sound, and those outer rows regulate what you want to hear and things like that. So that answers our question here of how energy is carried by sound waves and turns into mechanical vibrations, fluid waves, chemical signals, and action potentials for the things we hear. It also describes a mechanism of hearing, including the part of middle and inner ear rolls, and discussing hair cells producing action potentials in the cochlear nerves. Something I do want to focus on, just making sure that you understand. So these are going to vibrate, causing the fluid in here to pulse back and forth. That pulsing back and forth is going to move all throughout the ear, and it's going to cause this basilar membrane to vibrate whenever it causes which is going to be a start of the sound. And whenever it starts to vibrate a little bit, it's going to allow these hair cells to move, which will then allow the increase of potassium coming into the cell. Whenever it does that, the potassium is going to cause a depolarization, releasing a neurotransmitter for the brain, along with moving these pressed-in proteins over here, which is then going to cause a movement of this tectorial membrane, causing more waves reacting with this inner row here of hair cells, and then causing you to be able to hear sound. I know that was a lot, but keep with me. You are doing fantastic, as I know you got this. Next up, what we're going to do is we're going to explain how the eye focuses on light. Now, this is important for me because I'm slightly nearsighted. I like to be able to not wear my glasses when I'm sitting up in the front of class. When I'm sitting in the back of class, I have to wear glasses, sadly. Honestly, sometimes I do it when I'm in the front of class. But it's all just dependent upon how I'm doing that day, what I'm feeling. So let's go ahead and explain that. We're going to mention the role of the ciliary body and suspensory ligaments of the lens surrounding the lens, and we're going to describe the different photoreceptors, how they convert light into electrical energy, and we're going to explain the on-center, off-surround, off-center, on-surround, and how they cooperate with visual fields. So this is our last little description that we're going to talk about. First, let's go over a little bit of anatomy of the eye. So right here, you're going to have your aqueous humor filling this anterior cavity, so that way the light isn't going to be bent nearly as much. But down here in this posterior cavity, you are going to have vitreous humor all up in there. So just fun fact there. All right. So whenever light starts to enter into the eye, it's going to hit your eye, and then it's going to move through your lens, which your lens will then refract the light more, so that way it can land on your retina, which is back here, and at the appropriate amount and at the appropriate convergence. Up here you have your optic discs, and nerves will run through here, which will then react with the retina and the different action potentials that are running through here, and we'll run through those action potentials in just a minute. So with this being said, we have our rods and cones over there. We have rods here. We have rods here, and in our fovea, we have a lot of cones. You have a lot more cones in your fovea than you do in your rods. But altogether, you have about 120 million rods and 60 million cones. So just keep an eye on that. Here you're going to have your photoreceptor, then you're going to have your bipolar cell, and then your neuron. Behind the neuron, you're going to have your blood vessels, which provide nutrients to the neurons. All right, now that we went through a little bit about that and how light is being bent in order to reach the certain part of the retina, we're going to talk about these muscles right here. So right here you have suspensory ligaments, and down here you have your ciliary muscles, all these little blue parts. You have them both all around your eye. Just imagine as if your eye is circular in this photo, if you think about it like a 3D object. So here your lens can do a couple things. Your lens can either get flattened, as we see here, by the tightening of suspensory ligaments, or it's going to bulge, which is going to be by the loosening of suspensory ligaments. So right here we're going to see our suspensory ligaments as you see them in black here, and then your ciliary muscle as you see in green there. And this here is your lens. So the ciliary muscle, when it contracts, is going to contract inward, not outward. Don't get that confused. Whenever it contracts inward, it's going to loosen your suspensory ligaments. So this is contracting. Whenever it relaxes, it's going to tighten suspensory ligaments. So let's go back to what we were talking about here. If you're contracting your ciliary muscle, which means you're loosening your suspensory ligaments, what are you doing to the eye? That's right, you're bulging. Now, if you're relaxing your ciliary muscle, which means you're tightening suspensory ligaments, what are you doing? That's right, you are straightening out your lens. So that will allow light to be able to be diffracted more or diffracted less. And then another thing that I just want to cover with you real quick is convex versus concave lenses. Whenever you have something convex, light is going to come this way and it's going to converge. While if you have something that's concave, it's going to go more out. So as we see with our lens over here, whenever it's bulging more, it's going to be more convex. So therefore the light is going to come closer and closer as it bulges. But with it flattening, it's going to become further and further out. Hopefully you never have a concave lens. That would be really bad. All right, with this being said, those who have excuse me, sorry. I think I already wrote this out. So those who are nearsighted, aka myopia, are going to need a convex lens. So that way it can shorten the distance because those people will need to be able to see see that appropriately. While those who are farsighted is the opposite. Blurry vision occurs whenever you see two dots right here and right here, not on the retina. Not on the fovea. It's going to hit just on the retina back here. This whole thing is the retina. So it's going to hit like right here, and that will cause blurry vision. Something fun about myopia is if you look through a smaller hole, it causes the light to hit more of the flat part, so that way you can see the image that you're looking at through that small hole better. Next thing we want to do a little bit is we want to talk about the photoreceptor, bipolar receptor, and the neuron that will take the response to the brain. So first thing first, we're going to talk a little bit about the molecule 11-cis-retinol. This is what it looks like right here, and it's going to have that cis bond, aka which light will be able to break sometimes. And along with this, just so you know, is retinol is also vitamin A. So we need a lot to make it. And going back to this light breaking that cis bond, is if that bond breaks, it's going to change the 11-cis-retinol to 11-trans-retinol, which looks like this over here. And so over here, you're going to have a GPCR, over here in our photoreceptor, this molecule is going to be in this GPCR right there. So whenever that 11-cis changes that, it's going to cause the activation, is changed to trans, it's going to cause the activation of this alpha subunit that is then going to cause the activation of PDE, which is then going to break up this cGMP right here. With that cGMP broken up, that means you're not going to have a working voltage-gated sodium channel right here, which means then you're not going to get these positive signals coming through the cell, and you're not going to have this voltage-gated calcium channel working. I'm just going to erase all those because I'll come back to this. And then that means you won't have glutamate coming into the cell. Then you won't have reaction of this GPCR, which means you won't close the TRPM1 channel. This TRPM1 channel, the calcium will then be open, allowing it to go into the cell, which will then bind to these vesicles, allowing the release of glutamate, which will then react with AMPA, allowing it to open, and cations to open into the neuron, allowing you to be able to perceive light. Now, if no light comes in, so I just talked about perceiving light. If no light comes in, we're going to see exactly that happens here. This isn't going to work, which means this isn't going to activate PDE, which is phosphodiesterase, which that cGMP will then bind to that voltage-gated sodium channel, which will then allow more positives into the cell, causing a BWONG, which I love that word, by the way, which is then going to allow this voltage-gated calcium channel, ligand-gated sodium channel up here. I apologize for that. But it's going to allow this voltage-gated calcium channel to open up, causing the exocytosis of glutamate, which will then cause the GPCR to be active, releasing activating this alpha subunit, which will close this TRPM1 channel, which means no glutamate will be released, as we see here, and meaning AMPA will not be activated, which will then not allow action potentials to go down to the brain, so we see and perceive dark. So these are with rods, cones, and things like that we end up perceiving. So I just drew what light looks like and what dark looks like. Light has glutamate down here, but not immediately after the photoreceptor. And then dark has glutamate here, but not after the bipolar cell. Alrighty, now that we've talked a little bit about how the eye is perceiving light, we're going to go a little bit into on center and off surround. So here we have our photoreceptors, our bipolar cell, and our neuron. With this being said, I need to fix something on here. Alrighty. With this being said, whenever light on our on center off surround, we're going to have mGlur6 as our GPCR. But over here with our off center on surround, we're going to have our AMPA channel in this bipolar cell. Now, going over about real quick what this off center on surround means is off center means whenever light hits the center, it's not going to have a signal. On surround means whenever the light hits the surrounding cells, it's going to give a signal. Now, tell me which one of these are going to give a signal to the neuron and which ones aren't. That's right. On center, whenever hit by light, will give a signal to this neuron down here, while the surround will not allow you to receive a signal down here. Another thing to also understand are these horizontal cells over here. Whenever light hits, it's going to not allow glutamate to be released. With that glutamate not being released, it's not going to activate this AMPA channel that is here, which means it's not going to cause a release of GABA, which is a neurotransmitter, which means GABA will not inhibit the chloride potassium channel, which means that it will release more glutamate into the cell, which will react with the GPCR, which will then close up this channel and not allow that channel to work, which will then not allow the release of glutamate into the cell next to the neuron. While when light hits the center, it's going to react with it, not allowing the release of glutamate outside there, which will then not allow the mGlur6 to be active, which means that tryptophan-1 can allow cations into the cell, which means that the calcium will then come into the cell. The tryptophan-1 is a calcium channel. The calcium will then come into the cell and allow the exocytosis of the neurotransmitter glutamate, which is then going to react with the neuron. Now, with the same thing, we just see something opposite happening down here due to this AMPA channel change. Whenever the light hits the cell and no glutamate is released, it's not going to activate the AMPA, and the AMPA is going to allow that calcium to enter into the cell, causing the exocytosis, so we won't see light whenever it hits the center cell. But when light hits the surrounding cell, it's not going to cause glutamate in here. It's not going to cause this AMPA channel to activate. It's not going to cause the release of GABA, and it's not going to not going to inhibit that. So that means these are going to work, and that means it's going to cause more release of glutamate, which means the AMPA channel will then go through, and then that will cause the exocytosis of glutamate in this cleft right here, causing the action potential to go with the neuron right here. Alrighty, so that explains a little bit of on-center, off-surround. So let's go over some of the rules real fast. So light always causes less glutamate release from the photoreceptor. The difference with it hurting the neuron is going to be due to the receptor on the bipolar cell, whether it's mGlur6 or AMPA. Receptor fields are named by what light does to the bipolar cell. Is it going to cause it to be turned on, or is it going to cause it to be turned off? So that's where we're getting our on or off. Next up, just a fun little fact, is the frequency of action potentials determine whether it is dark or light. So knowing your light is going to pick up on how many action potentials there are, and it is going to allow you to see more or less. And something else I wanted to talk about a little bit is these receptor fields we're talking about. I'm going to draw a little bit about these on center, off surround, which this is an example of your surround and this is an example of your center. So I'm going to draw that right here. We're going to make one light and we're going to make this dark. And we're going to make this dark and this light. Whenever you see it light in the area, that means it's going to be on. Whenever you see dark in the area, that means it's off. So that means this is off center, on surround. This one is on center, off surround. All right, now that we clarified that a little bit, I just want to teach you that the surround is going to take up 40%. It's going to take up 20% in this side and 20% in this side, while your center is going to take up 60% of the action potentials that are sent. So let's say that you have light coming into the cell and you're hitting this much of the cell. How many action potentials are you going to have? I'm just going to separate this into 2 action potentials, 6 action potentials, 2 action potentials, just for us to be able to understand a little bit. So we know this on center, off surround. So on center is being hit by light. What is that doing? It's causing action potentials. So therefore, we need at least 6 right here. So 1, 2, 3, 4, 5, 6. Let me just draw these bigger. All right, so we have 6 action potentials happening right there. Now the light is hitting this right here, which means that is off. So that means you aren't going to receive 2 there. But light isn't hitting right there. So that means that one will send action potentials, meaning you're going to have 2 more. So you're going to have 8 action potentials right there. Now let's do the same thing. Light is going to cover the same amount. It's going to cause, you have your 2, 6, 2. This one is on surround, so that one's not going to be on. This one will, and this one won't because it's off whenever light hits it. So you're only going to have 2 action potentials coming from there. So there's a little blurb on there. And let me just explain a little bit about Herman's grid. So I can't really draw Herman's grid. It wouldn't quite give you the same effect. So we're just going to look it up real quick. I'm going to go to an image over here. We're going to look at Herman's grid right here. So let's say you're going to look. Oh, I can't draw on it. That's a little sad. Awkward. We're just going to snap. So whenever you look in a corner, such as the top left corner, the other boxes through your peripheral vision look, the other intersections of the white lines, they look gray. If you're focusing on that top left corner, the other intersections look gray. But then when you move, you are going to perceive that intersection to be white and the others are gray. Why is this happening? Well, let me show you. You have your macula and your... Let's see if I drew this out. I'll draw it out for you right now. So you have 2 different types of receptor fields. You have your macula and you have your periphery. So your periphery are larger receptor fields. Your macula are smaller receptive fields. So let's explain what's going on there. So you have your Herman's grid whenever you are focused in an area, we're going to do red equals focused. That is going to be your macula. That your receptive field is smaller. And so you're going to see more areas like this. So that way you can perceive the light better in that area. Now, we're going to do blue as your periphery field, as your less focused area. Periphery. And that receptive field is larger. With that being larger, it's going to pick up on these darker spots right here, and in the corners of these, which will then send actin potentials that area is dark. So that is why you're going to see more of a gray area right in here. Let's see. More of a gray area right in here, because you're picking up on these action potentials that you're receiving from these corners of these boxes right there. And so that is how the Herman's grid is working on you. I hope you enjoyed this little session. Now that we've covered all these topics, make sure that whenever you eat something such as cheesecake, you're like, man, my ligand is really activating my IP3. So that way, your wife will love you so much more. And if you don't have a wife, hopefully you can get one, because everybody knows women love to hear the science that you tell them. Or if you're a woman for this, men love to hear you just explain how things work. Men are so used to it that whenever you do it, it sounds so much better. Women typically refer to this as mansplaining, which everybody knows, both men and women alike love it. So go ahead and do this. Just kidding. Don't take my advice on that. There's other advice that I could give, but not in this video. So I hope you have a great rest of your day and you enjoyed this video that fulfills all of our questions here. Thank you.